Hello everyone, welcome to today's webinar brought to you by Microbial Insights. This is on can we use microbes for this? So bioremediation for the next generation of contaminants. We appreciate that you are taking the time to join us today, and it is my pleasure to introduce our esteemed speaker, Dr. Kevin Finneran. Dr. Finneran is a professor of environmental engineering at Clemson University. He received his PhD in microbiology in 2001 at the University of Massachusetts at Amherst. He has been a professor at the University of Illinois, Urbana campaign, and a consultant at Geosyntec. His expertise in environmental remediation using combined microbial chemical reactions. He is a fellow Kavli Fellow of the National Academy of Sciences, and he owns and operates Finneran Environmental, which supports on the most difficult of or problematic environmental remediation projects. So now, before I let Dr. Finneran take it away, I would like to remind you all, as always, this webinar is being recorded and it will be available in the resource section on Microbial Insights website, hopefully by the end of this day or by the end of this week. So don't worry, we'll also be sending out an email with a link and certificate of attendance following this webinar to those that have registered. There's also an included informational handout you can look at during this webinar. It's in the handout section of your sidebar. So after he is done, there will be a little bit of time for questions. So feel free to type your questions as the presentation goes on. And either I or Dora will try to read off as many as we can so that way you can get your answers. So join us in welcoming a true advocate for sustainable solutions, Dr. Kevin Finneran.
Marshall, thank you so much. Dora, thank you so much for having me. Sam, obviously for the invite when we saw each other in San Diego. For the audience, just to kind of tee up the expectation, this is going to be a relatively short and light presentation. Reason being, this is a presentation that I gave in part, tweaked it a little at the San Diego version of the Association of Environmental Health Sciences conference. So if you go to the UMass conference on the East Coast, AEHS, that's the West Coast version. And I was invited to give a lunchtime talk, which I'd never given before. And the only rule that they gave me was keep it light. And I was like, okay, so we'll keep it light. And I came up with this topic of sort of thinking about what can we do with bioremediation? What are the contaminants we might be tackling once we kind of wade through PFAS right now as the topic that is keeping us all busy. And given that I can give this broad perspective, I guess that really just means I'm getting old, but I can live with that. And hopefully after this, many of you are thinking about the same contaminants about which I'm thinking, which means that we'll have some good solutions teed up, microbial and otherwise, going forward. So with that, I'll kind of jump into my thoughts of what are the contaminants we can use bioremediation for going forward. So what I'm going to try to talk about is we all know where we are for fluorinated compounds and why I'm sort of not very hopeful about using microbiology as the primary means for remediation of PFAS compounds, even though that's the focus of where we are right now. But then talk about a variety of compounds and contaminants going forward, where I have a moderate amount of hope to a good amount of hope that we can deal with these compounds using microorganisms as primary means of remediation. And then hopefully this will kick off a discussion for those who feel like hanging around and asking questions online and in the dialogue boxes. So I'm a microbiologist and by training and by interest, and I teach a lot of microbiology classes, and I tell my students, microorganisms, cells in general, eubacterial, archaeal, and even eukarya, and they generally only do two things. Cells generate energy as ATP, and they build new biomass. And it's in this sphere that environmental engineers and microbiologists and remediation scientists have to play. I mean, there's a lot of moving parts to those different types of reactions, but really that's all cells are doing. So when we're using microorganisms to clean up contaminated environments, we have to ask, is there a benefit in terms of the energy, or is there something that can happen during biomass synthesis that we can take advantage of? And on the catabolic side, the energy generating side, most of these tend to fall into the oxidation reduction type reactions because redox reactions are what dominate in catabolic reactions. So electron donors, electron acceptors, and electron transfer becomes critical on that side. In terms of the anabolic side, building biomass, is there anything along the, say, lipid bilayer, the cell wall, or anything where we can take advantage of partitioning technologies or immobilizing contaminants or aggregating contaminants when cells build new biomass? So hopefully a lot of these contaminants we'll talk about today we can address, but we'll start right out of the gate with the one for which I don't have a lot of hope that we're going to use bioremediation, microbiology, cells, as the primary means to deal with this contaminant. And that's the one we're all in right now. So we're all in the PFAS world, whether we want to be or not. A couple years ago, Popular Press got a hold of a couple of stories, gave these things a really cute nickname, forever chemicals. Most people know more about PFAS and water and biosolids and wastewater than they know about what is the primary Superfund site close to their house with hydrocarbons or metals, metalloids, radionuclides, or chlorinated solvents. They feel the risk to these is larger than anything else, which is ironic as we all sit here on our waterproof chairs, near our waterproof curtains, coming here in our waterproof car seats, walking across waterproof rugs, and hopefully we don't have any fires in the house that we put out very easily with aqueous film forming foams, which work a lot better than water to put out fires. That said, we're afraid now of PFAS in water, biosolids, wastewater, wherever we might find them, airborne deposition even. And my lack of hope is not because I'm not reading good papers on microbiology in PFAS reactions. There are several papers coming out. It's just when I think about, again, what do cells do and why do they do it, I don't feel like relative to, say, the partitioning technologies, granular activated carbon, colloidal activated carbon, ion exchange, even some reverse osmosis, thermochemical technologies, microbiology is going to be used to deal with perfluorinated compounds. And this just comes down to simple bond energy. So, bond energy of a carbon-fluorine bond, we were hoping that what we know from carbon-chlorine bonds and reductive dechlorination might carry to carbon-fluorine bonds. But the bond energy difference is vast, irrespective of the electronegativity of the carbon-fluorine and carbon-chlorine bonds. On the left, we have carbon-fluorine bonds. And if anybody's a child of the 80s, like I am, you will immediately recognize that scene from the movie Predator. Arnold Schwarzenegger on the right, Carl Weathers on the left. Take a moment of silence again. Carl Weathers always was one of the best. And that's the bond energy of a carbon-fluorine bond. You're not going to get in there and break this bond easily. Whereas the bond energy of a carbon-chlorine bond is conducive to being broken by reduction or even oxidation. As such, we can use microorganisms primarily for carbon-chlorine bonds, and we've done that. Reductive dechlorination, oxidative strategies with co-metabolism, they work for carbon-chlorine bonds and chlorinated solvents. It's not carrying to the perfluorinated compound as well as we would like. There are good papers coming out every day on some microbiological tools, but relative to all of the non-microbiological tools, predominantly the partitioning technologies, I just don't have hope that we're going to use bioremediation for perfluorinated compounds the way we have for other things. That's not to say we don't have good technologies. It's just that this one, maybe the microbes aren't going to be our go-to with respect to this contaminant, this class of contaminants, and that's fine. And if I'm wrong, and I hope I am, kudos to the people or person who find the microbial technology that we can use readily. So then I started thinking from there, if we're stuck in PFAS land for a while here, trying to rip apart, Carl and Arnold's arms, what are the contaminants for which we can use microbial strategies? And I was thinking also with respect to PFAS, maybe we're just looking in the wrong place for the cells, because on the carbon-chlorine bond side of things, I was thinking, when dihalogenamonas was identified as a microbial genus species that takes apart carbon-chlorine bonds, they didn't find it in a chlorinated solvent contaminated site. They found it with grape pumice and grape waste, where there were just naturally occurring carbon-chlorine bonds, and the cells were evolving over a much longer time frame. Problem is, on this planet, finding organic carbon-fluorine is difficult. We've got to go to extremely hot places, black smokers, lava flows, earth core type places. These are hard places to sample. So again, when I'm thinking of where over evolutionary time carbon-fluorine bonds have been dealt with microorganisms, they're hard places to reach to figure out, can we use that microbiology? And then we might not be able to replicate those extreme environments in the environments in which we're finding perfluorinated compounds. So maybe we're going to be in PFAS land for a little while longer, but there's some other contaminants coming down the pike, many of which we're already probably thinking about, but for which I think we might have a place for microbial activity. If I think about the next big frontier of remediation, so once the PFAS boogeyman moves to the side and the next boogeyman comes into view, I think, this is purely opinion based on what I read, I think it's going to be microplastics. You know, PFAS will kind of start to fall to the background. Microplastics will become, oh my goodness, we should have been dealing with these much longer than we have. And some of us are already addressing them, and there's a variety of technologies, but I do think microplastics, I have a modest amount of hope that there is a microbial process that might help us with microplastics. Because one of the first steps in microplastics is aggregating them. The issue with microplastics individually is because of their size, filtration technologies are difficult. And because of the distribution of microplastics in surface water in particular, aggregating these plastics together makes them easier to clean than if we leave them distributed and sort of what planktonic in surface water. So then I think to myself, well, cells could have a role here, because one thing cells do extremely well when they're making new biomass is they form flocks and they form biofilms. So biofilms, right? Everybody knows this. This is the way cells grow on surfaces. There's protection to this. There's stability to this. There's resource management relative to growing biofilms. And cells signal other cells using chemical signals to come in and join these biofilms. And they release these extracellular polymeric substances to recruit new cells and sometimes other chemistries to build this biofilm. So this is on the anabolic side of the reactions here. When cells are making new biomass, maybe we can take advantage. These extracellular polymeric substances very often interact with organic material in the natural environment, dissolved in particular organic matter. So if I think of microplastics being out there in surface water, you know, floating around freely in planktonic, if we can develop biofilms in these environments, they will aggregate microplastics together, which might make them simpler to filter, to skim off the surface, to have them flocculate and fall to the bottom. Something we've been doing in secondary wastewater treatment, you know, for decades. Secondary wastewater treatment is dominated by forming flocculent material in the aqueous phase from growing microorganisms with carbonaceous BOD, and then it falls to the bottom as biosolids, and then we recover it. Can we do the same thing with microplastics in naturally occurring waters, whether that's a marine system or whether that's a freshwater system? So I have a modest amount of hope that when we go into this next frontier of microplastics, maybe microorganisms can have a role there. And that is an open area of exploration, both on the research and practice side. I know many of my colleagues in academia are already investigating this, and I've already seen a lot of good talks on the industry and consulting and practitioner side looking at this. Can we bring microbes into this? I think we can. And let's see where we go with this in the future. Another class of contaminants, this one we're all responsible for again, lithium and the rare earth metals. All right, we all have a computer in our pocket now. Not even a phone anymore. It's actually a much better computer than it is a phone every time I try to talk to somebody. I'm an iPhone person, not an Android person, but irrespective of that, they use these rare earth metals. And then obviously lithium batteries are going to increase over time as we electrify our vehicles. Now, the electric vehicle argument aside, I always say to my friends, if you're on solar and you have an electric vehicle, you know, you have quite a bit of moral high ground, and you can tell me anything you want to tell me about my gigantic Nissan Armada, which I still love and is quite inefficient. But, you know, as we electrify our vehicles, if you're not on solar, we do have to address the how do we generate electricity side of things. We'll leave that for somebody else. But lithium batteries are going to end up in waste. And a lot of, from our computers and our pocket computers, our iPhones and Androids, a lot of rare earth metals. Is there a role for microorganisms in helping us recover these? Because this is a finite resource on the planet. As we electrify our vehicles, lithium mining is going to ramp up. lithium mining is actually quite environmentally problematic. It's a really bad process for the environment. So can we recover lithium in a usable manner from the wastes we're generating? And do microbes have a role? Well, a group of microorganisms that I think are going to play a really important role are ones I've been studying since graduate school, those that are iron reducers. They breathe iron the way we're breathing oxygen to get their energy on the catabolic side of the reaction. So they've learned to access solid phase electron acceptors in natural systems, and in doing so have developed an array of different approaches to address solids, and in one case, solubilize these solids by releasing extracellular ligands or chelators. Geothrix type species, genus and species, they release an extracellular chelator. That's down on the right there. I don't know if I can actually make things work kind of right here. You can see it. Rather than reduce solid phase iron 3 directly, they release this chelator. The chelator interacts with the iron 3, solubilizes the iron 3 the way all chelators work with metals, like EDTA working with metals to keep it in solution, brings it back to the cell, and the cell breathes it that way. And the graph on the right was iron 2 recovery from a variety of systems in which we had filtrate from Geothrix fermentans, and the 100% filtrate of the biomass was the best at recovering iron 2 in solution from solid phase iron 3, meaning as the cells grew and generated more of this chelator, we could recover more iron in solution. The question is, can we carry this over to the other metals, particularly lithium? Is there a system in which we can microbially recover lithium from solid phase waste such that it's chelated and brought into the aqueous phase and then recovered through chemical techniques and chemical engineering down the road to recover it as lithium that could be reused perhaps in lithium ion batteries? Can we do that for the other rare earth metals? I have a good amount of hope that we can do this because to the microorganism, the solid phase doesn't matter. We just need to make the conditions conducive to the growth. And then hopefully the metals that we need to recover are going to be recoverable irrespective of the type of waste we find them in. So I have a good amount of hope that we can figure out some things for metal recovery. And there are organizations already working on this. You know, this is not necessarily something 100% new, but certainly needs a lot more work and look and research on both the academic and practical side before we have a way to do this and make it inexpensive enough that these recycled materials are going to be competitive with the virgin materials. You know, we all know that recycled paper is more expensive than virgin paper, and there's a reason for that. And the costs have balanced out now where recycled versus virgin paper is reasonable. We need to do the same things with these metals, and that's going to take some time. But I hope that we can get there with microbes. So jumping back into like from the small to the very large, a lot has been in the both peer-reviewed and non-peer-reviewed literature on mine waste again. It's funny, we're over here looking at PFAS in water, even though more PFAS exposures in our house or a car. And these mines are massive. These mining operations are hundreds of thousands of acres of land being used, and they liberate millions of liters of contaminated water with metals and metalloids in them, sometimes at extremely low pH. And luckily, we're starting to refocus our ideas that we want to clean up mine waste. And I think microorganisms have a big role in cleaning up mine waste, because a lot of mine waste remediation technologies start with, let's raise the pH of the water, irrespective of whether the metal and metalloid are redox active. By raising the pH of the water from that 1, 2, 3 of the mine waste up to the 6, 7, 8, we can precipitate metals. And then we can use partitioning technologies and standard chemical technologies. But chemically raising the pH can oftentimes be problematic because you chemically raise the pH and it comes right back down. Microorganisms, however, can shift the pH up, and they can do it over a longer time period. It takes longer, but then it stabilizes at higher pH, because microorganisms in the subsurface are what generally control the pH dynamics of the solid and aqueous phase in groundwater. And the reason being, if you look at the reactions there on the right-hand side, all of these are proton-consuming reactions. If we look at iron reduction, manganese reduction, sulfate reduction, all metals and electron acceptors associated with mining, because you use sulfuric acid to leach from the mines. You liberate iron, you liberate manganese. Iron reduction, manganese reduction, and sulfate reduction are dominant metabolic respiratory processes during mine waste and mine operations. So if you look at them, all of those consume protons. And in consuming protons, that raises the pH. It's slower than a chemical pH adjustment. But if you look at the graph, that's actually one of my sites at which first step we had to do in order to use reductive dechlorination and to get to chromium reduction was to raise the pH. And we used a combination of formate, lactate, and acetate at equimolar amounts to all available electron acceptors, iron, manganese, sulfate, and chlorinated solvents, and chromium. And over about an 18-month time frame, the pH of the groundwater in the contaminated zone went from 4.5 up to 6.5, 7. It's a slower process, but the stabilization keeps that pH up, where there was no rebound back down into the low pH. We stayed at pH 7 and then pushed it into chromium reduction and reductive dechlorination. At these larger mine waste operations, there's a lot of land at play, and a lot of this is on the surface. So if we can develop surface water mediated reactions for raising the pH slowly over time, we can stabilize perhaps the metals coming out, the copper and the antimony and the tin and the zinc and the lead and even the lithium, such that we capture it before it makes its way to the larger water bodies and then causes problems for the ecosystem, for fish health, for microinvertebrate health, and then obviously tertiary human health. So I'm very hopeful that as mine waste remediation becomes a larger topic, I got to go first time ever it's so long ago now, last October, I went to a mine waste conference, and the scale of these is just massive, but it still comes down to the baseline reactions in the water in the solid phase. So I'm really hopeful that we can start using microorganisms and bioremediation in these large mine waste operations, because the reactions to raise the pH are a first step, and microbes are very good at that. And then the last one, I get really excited when I see quinones, because anybody who knows me knows I love quinones as an electron shuttling agent. And it turns out there's a tire stabilizer that leaches off the side of tires, and in the Pacific Northwest, it's very problematic because the 6-ppd quinone compound, when it gets into surface water, apparently is very problematic for salmon. And in the Pacific Northwest, salmon is one of not only a fish that they're very proud of in their natural environment, it's a huge economic driver in the Pacific Northwest. So this 6-ppd quinone in the Pacific Northwest has become a very focused contaminant of interest. And that's a contaminant right there in the quinone form. Right here in the dead center, there's your quinone, those 2 carbonyl groups hanging off of that aromatic ring. And when I see that, I see a terminal electron acceptor in a respiratory process, because we've been looking at quinone hydroquinone couples for a very long time. So I'm very hopeful we'll be able to figure out something with this, because again, anybody who's known me, even back since I was a graduate student, on the left-hand side here is the first slide I ever used in any talk I ever gave when I was studying MTV. But quinones are terminal electron acceptors. If you look at microbial metabolism on the catabolic side, there is a quinone hydroquinone couple in the electron transport system. It carries both electrons through the electron transport system and protons from the cytoplasm out of the cell membrane to form the proton motive force. Quinones are part of microbial metabolism, part of cellular metabolism. So I'm hopeful that this 6-ppd quinone is just another terminal electron acceptor, the way here we're looking at oxidized versus reduced humic substances, or the model electron acceptor quinone, the anthraquinone disulfonate to anthrohydroquinone, because they're reduced to the phenolic form, and they cycle between these. And there are differences between these compounds in terms of how, first, foremost, toxicity, secondarily, how easily they can be partitioned out of a given environment, and third, can these be part of the remediation process themselves by recycling electrons that are part of microbial respiration. So while 6-ppd quinone is not maybe as ubiquitous a contaminant worldwide, nationwide, it's got a very specific focus, but I think there's a good role for microorganisms in any quinone-bearing molecule, and I'm really hopeful, and we're beginning to investigate this in our lab here, along with the other things you're seeing, like the microplastics and the lithium recovery. Again, anytime I see a quinone, I get excited because I'm hopeful it's a terminal electron acceptor, and we're really hopeful that the 6-ppd quinone won't be any different. and maybe we can use microorganisms to help clean up a contaminant that, again, is more of a surface water issue as it makes its way from, roadside hazard, overland, into surface water, and then ultimately groundwater where we can clean it up. So to conclude, I told you it was quick. I'm a microbiologist. I love microorganisms, believe in their power, but sometimes we need to acknowledge they might not be the primary tool we're going to use in environmental remediation. And I think the limitations we're seeing with perfluorinated compounds echo that point. But microbes surprise us all the time. And if we play in that sandbox appropriately, cells generating energy, cells generating biomass, we can make microorganisms do some of the work, if not all the work for us for a variety of these contaminants. And new chemistries are being generated every day. New chemistries to replace the perfluorinated compounds are coming out as we speak. And we're hopeful that those won't be problematic from an ecosystem, environmental, human health perspective. But the problem is you typically don't find out about that until they are problems. And then you have to be reactive rather than proactive in dealing with them. But along the way, microorganisms are going to be part of the solution. And with that, all of this information and knowledge that I just gave you came to me because I got to work with a variety of great graduate students over a number of years on a variety of topics that kind of brush the surface of all of these. So this is those students, past and current, right there. And with that, again, to Sam and Dora Marshall, thank you so much for having me. Hopefully we kept it light. And hopefully we can all get together at some point and talk about these contaminants other than PFAS one of these days. I know that won't be the case for at least the next year or two, but again, I'm hopeful. All right, thanks so much. I appreciate it, everybody.
Awesome. Thank you so much, Kevin. This is a great presentation, a good summary of quite a few topics, and it's exciting to think about the future of remediation and what's going on. So for those of you in the audience, if you have questions, can you please go ahead and type them into your GoToWebinar panel? And we'll ask the questions for Kevin right now. So Kevin, with microplastics, one of the questions that I have for you is there's so many different types of plastics and there's sorting and a lot of different intricacies that go into remedial design for these types of projects. Do you have any suggestions for people or kind of a broad overview for people as they start to approach this, like, experience of what you think would be good for them to do and to look at, and then, maybe some overall guidance on how to do that?
Sure. And this is mainly coming from my reading, going to presentations as well. So a lot of the people on the call might be people from whom I've learned things. It seems like a lot of the plastics that are derivatives of polyethylene integrate well with potential microbial solutions because we know a lot about how polyethylene, like HDPE type compounds, interact with microorganisms, mostly from the food industry where you're trying to keep microbiology out of the plastics or off of the plastics material. So there's a whole array of knowledge on polyethylene derived plastics and how microorganisms can or cannot interact with them. As such, we do know that as microorganisms grow quickly in the presence of polyethylene derivatives, they can aggregate, like even macro plastics down to the micro scale plastics together. And most of the work, again, has been done to keep this from happening in the food industry. But if people are starting to look at this, the polyethylene derivatives, and there are several, you think of like, right, the shredded plastic bags and the, type soda bottles that get shredded down, and it's those tiny little fibers that make their way into the natural environment and surface waters. Polyethylene type plastics do get aggregated by microorganisms already. It just comes down to, is there a way to engineer that in systems in which, you're going to have to control flow? You're going to have to capture flow with microplastics, funnel it through a truly engineered system, and aggregate the microplastics in an engineered system. This is not, right, there's no in situ technology for the open ocean. There's no in situ technology for rivers and streams into which there are already microplastics. We're going to have to harness real capture technologies along with this, but once captured, the data suggest that microorganisms can interact with polyethylene, at least derived plastics, and there are several classes that are in the microplastics realm. So that's where I'm starting, and that's maybe a place other people are starting, and because there's a lot of data already from the food and food industry.
Great, thank you very much. That's very helpful. One of the questions that came in is talking about PFAS. And I know like Peter Jaffe's done some really interesting work about partial defluorination, and Shailey Mahendra's group's done some work with fungal organisms. Can you talk a little bit about your take on the value of, our understanding of some of these at least partial pathways that are now available?
Sure. And again, I mean, lots of others. Natalie Capiro up at Cornell published a variety of papers on good microbiological interactions with perfluorinated. And so for everybody listening, I'm not saying there are not papers indicating microbes interact with PFAS compounds in a remedial type strategy approach. But when I think of like a remedial design in the field, do I think what is going to be the dominant technology and strategy we're going to use to clean up a particular site, whether it's engineered wastewater treatment facility or in situ groundwater? If I think of things like reductive dechlorination and chlorinated solvents, I immediately think, yeah, the microbes, we could probably go with those as the primary and crack them out. With perfluorinated, I think it's still going to be chemical technologies, partitioning based, thermochemical, photocatalytic. There's a lot of good technologies, energy intensive as some of them may be. And based on the data I've seen from the microorganisms, where I think there's a lot of utility going forward is when we have these concentrate streams from, like, say, foam fractionation, where we're going to concentrate the perfluorinated compounds, or those few environments like a wastewater treatment facility or a landfill where the concentration of the perfluorinated compounds is higher, right, away from those crazy PPT and sub-PPT levels. These initial reactions that we're seeing, particularly with precursor compounds, knocking them down to the smaller, low molecular mass perfluorinated compounds, the microorganisms will likely have a good role there, because microorganisms tend to do better at higher concentrations. And we know that a lot of these precursors can get knocked apart to perfluorinated compounds by microbial activity. There's where we might see microbes having some larger role in the perfluorinated compounds. And this is all, again, pure opinion. But when we get down to the PFAS, PFOA, Gen X type compounds at low concentrations, I just don't see the microorganisms winning versus, say, a partitioning based technology. But the good data are out there. And again, I hope I'm wrong. I mean, this is not one of those places where I'm rooting against the industry. I hope somebody comes up with a great microbial technology for PFAS and puts it in play.
Well, we would agree with you, Kevin. That's something we're hoping for too. It would be nice to have a good assay that people could use for this. And we haven't offered anything yet purely because of that. There's still a lot of research that needs to be done, I think, to understand it and see actually even if there is anything that can fully defluorinate. The next question that we have for you is, have you done any research on pharmaceutical compounds and degradation of pharmaceuticals?
Personally, no. I mean, only beyond reading, and then in this case, my reading is always limited to wastewater treatment facilities, digesters, and things like that. Yeah, whoever is asking this question, there's another niche for microbial activity, particularly in engineered systems, these pharmaceutically active compounds. I felt like before PFAS, they were coming into better focus, and then like PFAS came along and just knocked everybody's vision right to PFAS. There are some papers on, from what I've seen, engineered systems, digesters that deal with even simple ones like ibuprofen, which is typically a dominant waste in wastewater treatment facilities, can be dealt with using microbial transformation. And there are good strategies from what I've read. I think we just got derailed on that, like every other contaminant, because of perfluorinated compounds. Yet, if you believe what I see every morning when I'm watching the news, the only thing we have in this country are different types of medicines and pharmaceutically active compounds. We're going to need scientists honing that as all of these different pharmaceutically active compounds make their way into waste systems. I would say wastewater is maybe the bigger field for that, right? Because it's human waste first. This is how they typically get to the environment first. So if we can knock them out in a wastewater treatment facility, maybe they won't make their way to a natural environment. But then there's always landfill waste, where most dispensers of pharmaceutically active compounds now that are not pills or something like that are coming in these injectors, and they just get thrown out, not recycled. Well, clearly there's always going to be some residual of that contaminant in that, and that's going to be a landfill issue. But yeah, whomever's asking, that's another place where I think microbes can play a good role, and there are good papers on it, but PFAS came along and just was like, oh, let's stop looking at that now.
Yeah, PFAS has kind of dominated our mindset for a few years now. So one of the other questions that were submitted was, when you think about parameters that you use in site assessment, can you talk about some of your favorite parameters that you look to see if microbial communities are healthy? And, you know, are you looking at byproducts of some of the contaminants that are being degraded? Like, can you talk a little bit about kind of how you view microorganisms?
Oh, yeah, that's great. And first and foremost, every site is its own unique animal, whether it's an engineered system like a wastewater treatment facility or digester, or a natural system with contaminated groundwater. You have to address the site as that site. You can use what you've learned, obviously, from other sites and other facilities, but do try to focus on that. I kind of go in first with the basics, which we all probably do. You know, you take your pH of the system, the most oft overlooked yet very controlling factor of the site, because that's protons in solution moving into and out of reactions, dissolved and total organic carbon that may be present, you know, and also now part of the partitioning dynamics. So I look at solid phase as well as aqueous phase carbon, because for these partitioning based reactions, how contaminants move into and out of the solid phase is almost as important as what happens once the microorganisms start interacting with it. If it's say catabolic reactions, we look at electron donor, electron acceptor dynamics, what hydrogen, low molecular mass organic acids and alcohols are present, are there any dissolved carbohydrates in the system before we even think of amending them, and then on the electron acceptor side, what terminal electron acceptors are present other than the contaminant, if the contaminant itself is a terminal electron acceptor. Will pH shift interact with the contaminants, akin to what I was discussing with the mine waste? And then with the microorganisms, diversity indices and matrices, I like to see environments that start out relatively diverse, right? Because you think about remediation, you typically whittle down towards the microbial communities and populations that you want to stimulate. But to get there, very often you have to start with a very good pool of microorganisms. Are there fermenters present? Are there acetogens present? Are there metal reducers and sulfate reducers present? Do we need the methanogens, and if so, are they present? So as we screen the microorganisms, is diversity starting out high enough that if we start to whittle down towards specific reactions, will we have that initial diversity, right? It's like we're making a soccer team. You want the greatest number of people on the field to do all the positions before you whittle it down to the 11 players you put on the field. If everybody comes and says, I want to be a striker, you don't have a team. Nobody's going to win the games that way. You need everybody on the field that can do the different positions. So from a microbial health perspective, you know, you think of the tools you have available at MI, and you want to know what is the initial level of diversity versus the specific organisms you're going to try to stimulate. And then from there, just, you know, the realities of geology, hydrogeology, like if we need to put something into the ground, are we going to be able to distribute it? Or is the groundwater flow and the geology so tight that we can't distribute, and therefore do we have to target more appropriately at the specific location where the contaminant lies? And then the last thing I always think about is it better just pump this out of the ground? I mean, you know, ex situ technologies are still very good technologies. For some reason, people don't want to use them, but sometimes you just got to go in and get the water and deal with it on the surface. So that's sort of like what I go through in my head when I'm helping people design remediation systems. And, you know, and there's a lot of other data we'll go after, but that's on a case-by-case basis.
That's a great summary, Kevin. Thank you. And just for those of you that are on the webinar, we uploaded 2 handouts, one on next generation sequencing, which has some of those diversity indices that Kevin was just talking about, and then a qPCR assay that we have called Quantarray that does like biogeochemistry. So you can look at a lot of those functional capabilities, redox conditions, sulfur cycling, nitrogen cycling, that kind of thing. So those might be helpful to you as you're doing an assessment. Okay, so Kevin, the next question, we have 2 questions that are kind of similar, and the first one made me smile. It says, you open this door by showing Arnold and Predator. Should we be concerned as humans about introducing microbes to break down contaminants? And he says, as a field technician, he's wondering, what do they eat after the contaminant is consumed? And then there's kind of a second question that's similar, that asked about, are there ways to isolate microbes from polluted environments that may be doing the remediation so that we could bio-augment into other locations? So if you can kind of give your opinion on bio-augmentation and that approach, that would be great.
Sure. So to the first point, and yeah, I mean, I appreciate that you noticed Arnold and Carl too, and Predator, one of my favorites. Got to keep life fun. I myself am not concerned because the vast majority of the microbes, first of all, that we use in remediation are not pathogens. So in terms of human health, there's a limit to how they could interact with humans or animals in a problematic manner. But, with all caveats that anything is possible in microbe life. But what keeps me hopeful is even in these situations, these microbes are very specific in their metabolism when they interact with contaminants as an electron donor, terminal electron acceptor. And typically, we see populations and communities associated with remediation increase, then decrease, whether we've added it via bioaugmentation or they grow as part of the naturally occurring community. So as the contaminants become limiting in these environments, the microbes often slow down their growth, then they die off, and then their biomass, number of cells per unit volume or mass decrease. So they're kind of coming and going with the contaminants, and they never build up to such a high biomass that they typically outcompete the other native microorganisms that are present growing on the naturally occurring carbon that is in groundwater or surface water, or certainly in like a wastewater system. Like for pharmaceutically active compounds, the ones that can knock out the packs, they're going to be a much smaller population in a digester, say, than the fermenters and the acetogens and the methanogens. So once they come and go with the packs, they're going to get knocked down by the other cells that are present. The second one, my graduate students, as part of any project they do, I say always try to get a cell or a microbial community, a mixed culture, because it's fun. Going after cells for bioaugmentation is fun. And we're doing a site, actually an aerobe. We just got a styrene degrader because of our site in South America has styrene, ethylbenzene. So we actually just isolated an aerobic microbial consortium, and it was fun, but it's difficult depending on the metabolism after which you're going. So aerobes growing on big hydrocarbons typically is not difficult. What I would think is to what I said about the perfluorinated compounds. Where are the organisms naturally occurring rather than where is the contamination? Because the microbes have likely been around a lot longer than these contaminants, and you want to go after the metabolisms that have been in place for carbon, fluorine, carbon, chlorine, hydrocarbons, metals, metalloids, and radionuclides that are not necessarily there just because the contamination is there. They'll be selected out by contamination, but again, dehalogenamonas, which is our other big dechlorinator, the dehalogenamonas that was first isolated was from a grape waste that had naturally occurring carbon chlorine compound in it, not chlorinated solvents. So I think it's about where do we go to get our initial inoculum associated with these compounds, and then how you coax them out for bioaugmentation over time. I can only ask patience. Like my graduate students think these cells are going to grow, they're going to have a culture in a month. No, it takes years, and then to refine it to a usable, repeatable culture that you can inject, very difficult. I mean, and some of these cultures are pesky. They're like houseplants. They die much faster than they grow. So patience and picking the right environment from which to get them.
Okay, so you talked a little bit about 6-PPD. Do you know if there's more research that's been doing, or are you doing research alongside these tire manufacturers to help us to address this issue? I know there's been a lot of publicity with streams and surface waters and the impact that it's having on fish. So just curious about that.
Sure. We're starting research. We haven't cracked the code yet by any stretch. Very new in the research, and we've got to get funding, more funding. And yes, the tire companies, right? Like Clemson has a great automotive engineering program, and we're trying to make inroads with Michelin, which is a South Carolina-based company. But Michelin, they like to make tires. They don't like to worry about what's coughing off the side of their tires as much. No knock on Michelin. They care about the environment, but it's, that's not the big draw. Most of the papers I've read thus far are on the toxicology of this and the fate and transport of this. And that I've seen thus far, I've not seen data on biotransformation using the quinone hydroquinone couple, which is what we'd be going after. But there's definitely interest broadly in what available technologies from can we prevent the 6-ppd from actually making its way and transforming to the 6-ppd quinone, because right, it scoffs off the side of these tires and makes its way over surface water, or do we deal with it once it hits the receptor? But that I've seen in the literature thus far, no microbial strategies for dealing with it yet. I and a couple of colleagues I know who really love this whole electron shuttling thing are starting to take a poke at it, and we're hoping we can generate some funding interest from the big tire folks, because I just bought 4 Michelins last year and I know they have the money.
Yeah, that's a good point. What about trifluoroacetic acid? Have you done any work with that compound, and do you know if there's hope for remediation?
Yeah, so well, I've not worked with that personally, but my colleague here that all of you know, Dr. David Friedman, who just stepped down as chair after, like 10 years being our chair, I know he's looked at that compound, and I know that he and several of his graduate students have presented and published papers on that compound, both being oxidized and liberating fluorine as fluoride, and I think it's more oxidation than reduction, but I believe the fluorines are reducible in that compound, and he and others have investigated that. So I personally have not, but I've seen papers through David and being on some of his committees, because he also studied for a long time the chlorofluorocarbons that, kind of sit alongside of all of this as well in the carbon chlorine, carbon fluorine world. So if you're interested in that, David Friedman here at Clemson would know immeasurably more about that topic than I would.
Okay, one last question for you, Kevin, and we just truly appreciate your expertise on all of this. One of the concerns when you're doing degradation of compounds is if you make a byproduct or another compound that is more dangerous than the original contaminant. And I guess in my head with this question, I'm immediately thinking of those perfluorinated compounds and ending up with cis, DCE, or vinyl chloride. Do you have recommendations for people on how they can be protective in their remediation goals to make sure that they don't end up with a more dangerous daughter product?
Yeah, no specific magic bullet like, oh, you can avoid it if you do this. My head goes to vinyl chloride immediately too, right? You got this one little link in that chain that's more problematic. Or if you think of like arsenic as a metalloid, like arsenate is bad, but arsenite is worse. This one is going to be non-site. Be honest with yourself with respect to when you look at a site assessment or you're doing a laboratory study. If you're generating more of these intermediates that are problematic in a laboratory setting, don't say like, well, in the field, we'll probably just leapfrog that. You're doing lab studies for a reason. If your lab studies are generating more vinyl chloride than anything else, you either one, need to change what you're doing in terms of electron donor, pH, bioaugmentation, and circumvent it that way, or that is just the dominant product. So when I do a site assessment and people come to me and ask, like, is bioremediation a reasonable tool here? If we're making those problematic compounds, like in the perfluorinated world, if we're predominantly taking these fluorotelomeres, knocking them apart and making these compounds that are more difficult to degrade, maybe you don't want to stimulate biological activity in that case, and you want to immediately use an approach that's based on chemistry or something else. If vinyl chloride is your dominant product in the field and in a lab, maybe bioremediation via reductive dechlorination is not the approach you want. And that's not to say you couldn't say like, hey, what about aerobic co-metabolism? That can knock out cis and TC and circumvent VC altogether. Maybe you just need to change your approach. So it's about doing the right upfront data analysis, which everybody's alluded to here, but you really do have to be honest with if this is what I'm seeing in the field and in the lab particularly, maybe this particular strategy is not one I want to go down this path, and I want to now go to an alternate strategy of technology, microbial or otherwise. But that's best I can do, because yeah, we've all been there when it's like, man, and I guess it's right, like a doctor, first do no harm. As an environmental engineer or microbiologist, don't make the problem worse.
I agree, very much so. Safety first, and that's a great one to end on. So once again, thank you so much, Dr. Finneran, for presenting this. And if y'all have any additional questions, you can just email info@microbe.com, and we'll gladly either forward that over to Dr. Finneran or answer that ourselves. And we just want to thank you so much for presenting.
You're welcome. It was a pleasure. I appreciate you guys having me.
Of course. Take care, everyone.
All right, bye-bye.